It is my great pleasure to introduce today's PI speaker, Dr. Antoine Molaro. Antoine's love for evolution and epigenetics started really early. So as a grad student in the lab of Greg Hannon at Cold Spring Harbor, he studied the evolution and inheritance of epigenetic reprogramming in mammalian cells. He then did his postdoc with Herbert Malik at Fred Hutch. And here he studied genetic conflicts in the context of chromatin evolution. Very recently, he started his own research group at the GRED Institute in beautiful Clermont-Ferrand in France. I learned how to pronounce that from Antoine. And Antoine is currently looking for postdocs. So if you enjoy his talk today and his work and you want to work in this beautiful place, do get in touch with him. Now, before I transfer to Antoine, I did overlap with Antoine for several months at the beginning of my postdoc in the Malik lab. So I'm sure Antoine's talk will be wonderful, but I can tell you a little more about Antoine in addition to his science. Antoine is a musician and he has kept that passion alive throughout his academic career. He's always enthusiastic and will tirelessly discuss science, music, and almost anything else at any given time of the day. So I highly encourage participants, especially trainees, to stay back for the speaker chat afterwards. It's sure to be excellent. And as Christine said, just send us a message in the chat box if you want to stay and we'll upgrade you at the end. All right, with that, take it away, Antoine.
Wow. Thank you, Pravuta. I was not expecting this personal. I'm gonna cry. So thank you all, and I want to spend a few seconds to really thank the Fragile Nucleosome community. I've been an avid listener to the seminars on YouTube after I put my kids to sleep or when they're on vacation at their grandparents', which is always a little bit of a break for us. And it seems like yesterday, Pravuta and Christine were explaining to me what Discord was. I can't really tell you that I got better at it, but I think it's a great tool, and the mentoring program is also great. So with that being said, thank you for this kind introduction. Thank you for inviting me. I'll tell you today about quite unusual histones and histone variants in particular, and how our interest in those histone variants sort of took us along the way into the study of parental conflicts in mammals. And so I'll tell you about that. As Pravuta alluded to, I just started my own independent research group, and it's been a very busy year, if you will, for me to move across the world and hire those great people that you see here. We study evolutionary epigenomics and genetic conflicts, and I'll tell you a little bit about that. This is a picture from us from this summer. You have staff scientists, Marine Fraisse and Joris Mordier, as well as students that have been here or are still here this summer, Guillaume Buisson, Valentine Petiot, and Nassim Abidar. I want to mention also, since there's still time now, my funding. We have startup funding from the Fondation pour la Recherche Médicale, and I also received support from our beautiful institute, the Genetic Reproduction Development Institute in Clermont-Ferrand, and the INSERM. So the lab is broadly interested in understanding chromatin regulation and overall epigenome regulation in the context of genetic conflicts. And these conflicts arise when co-evolving genetic entities engage in antagonistic interactions. Now, because this is often triggered by opposite evolutionary interests between those two parties, it will trigger adaptation and counter-adaptation cycles over a very short evolutionary time. And these leads to what we often refer to as evolutionary arms races. This is sort of a crash course in genetic conflict in one slide, but I want to mention that these are not just isolated examples of evolutionary arms races. We have such genetic conflicts can shape very fundamental biological processes at the level of genes, at the level of chromosomes, at the level of whole individuals. So we are particularly interested in studying those evolutionary negotiations in the context of chromatin regulation, more specifically in the context of chromatin modifying enzymes or histone variants. For this, we almost universally use phylogenetic approaches to uncover the hallmarks of those genetic conflicts in those pathways. And then we go in vivo, in cell culture, and in mouse models to study the consequences of this accelerated evolution or those evolutionary arms races on the epigenome. And more often than not, this leads us to the germline where genetic conflicts are most intense because they influence inheritance and have a profound effect on speciation, for example. So more often than not, we end up studying those pathways in the context of reproductive cells or the early embryo. So that brings me to the topic of my seminar today, which is unusual histones mediate parental conflicts in mammals. And I'm not gonna say anything very novel here to this crowd, but to tell you that histones are at the heart of chromatin structure. They organize the nucleosome and they organize DNA in this context. There's 4 flavors of histones, H2A, H2B, H3, and H4. And in addition to those core histones, decades of research have shown that standalone genes can encode for histone variants that can replace those core histones in various chromatin contexts. Now, when you think about histones, you think about old proteins, ancient proteins, proteins that originated sometime in the last common ancestor to eukaryotes, with related proteins even outside of eukaryotes. And it might be no surprise to most of you that the current paradigm assumes that histones and most of their variants perform a sort of universally conserved functions. And so we got interested in a specific class of histone variants known as short H2A histone variants. And these variants replace core H2A in the context of mammalian germ cells. And so we got into this project originally by wanting to understand their evolutionary trajectory, which I'm going to mention to you right now. Well, I want to mention that this work has been, many labs paved the way for our understanding of short H2As from a structural basis, and this is something I won't talk about today, but from a true sort of germline perspective, many labs have contributed to this work, and I want to mention them here briefly at the bottom of this slide. So now going back to the evolution, we got into this project by wanting to study the evolutionary trajectory of short H2As. So what I'm showing you here is the maximum likelihood phylogeny of the histone fold domain of H2A and its variants. And you have the topology here that is supported by bootstrap values that I highlighted here in black. You have your favorite H2A and variants that you probably know and love. And really what I want to highlight in this portion of the tree is the fact that those histones are closely related to one another, and the terminal branches here are sometimes barely visible, indicating the strong conservation of those histone sequences. So unlike H2A and most of its variants, short H2As belong to 4 phylogenetically distinct clades of histones. So that was the first finding: H2AB, L, Q, and P. And unlike most H2N variants, which have ancient origins, short H2As originated quite recently in the last common ancestor to all placental mammals. They're almost universally linked to the X chromosome, meaning that they're encoded on the X chromosome. And one of the very interesting and sort of most surprising finding was that short H2As are subject to unprecedented levels of diversification. This is quite surprising for such proteins. This is something that I want to illustrate further here. If you look at the amino acid identity between H2A proteins, between species that are separated by that many millions of years here. For example, human and pig are separated by 94 millions of years of evolution, and yet their H2A sequence are very close, over 90% identity here. However, when you start to plot a short H2A identity in this graph, you find that they fall below this line very quickly. And we can further show in this publication that I highlight here at the bottom, we further showed that this was a consequence of diversifying or positive selection in some of those cases. So again, a very surprising finding here. I mean, these are histones we're talking about, not just immune genes, for example. So one important aspects of short H2A's biology, in addition to their evolution, and this is something that of course was interesting to us from the beginning, was the fact that they are abundantly expressed during spermatogenesis. At the onset of meiosis and during spermatogenesis, short H2A's are incorporated into chromatin. And this is a result, again, of a lot of preliminary work from many groups here. So the hypothesis about their function was that they must be playing some important role in the process of spermatogenesis or at the exiting meiosis, for example. So we investigated this question by using a mouse model approach. And what I'll tell you today is our results knocking out a whole family of short H2As, in this case, H2AB in the mouse. So we used, in collaboration with Janina Farms, we used CRISPR-Cas9 technology to induce frame-shifting deletions in all three paralogs of H2AB in the mouse genome, on the mouse X chromosome. We backcrossed those, what is essentially triple knockout mouse models into a pure black 6 background for several generations. And this is what I'm going to tell you about today, even more generations as time flies and I reestablished the colony here in France. So of course, the first obvious question was to say, what is the fertility of those knockout males? So what I'm showing you here is a measurement of this fertility in the context of a fertility assay. These are wild-type black 6 females crossed to wild-type males. You can see here the individual live pups in individual litters that came out of those crosses. Average litter size in wild-type is about 7, that's it. And our first surprise came when we looked at knockout animals because they were pretty much fertile just as their wild-type counterparts. Here I'm showing you knockout males crossed to wild-type females, and these are sibling males to those wild-type males, just to give you an idea of the genetic background. So that was a bit surprising, and I want to briefly mention here that this was also seen by another group that knocked out H2AB. And again, they saw a very mild, if any, effect on fertility. So sort of two surprising results here. So now we have a conundrum. We're like, well, okay, so H2AB has been around for over 100 million years in placental mammals, only very few species. Lost H2AB, maybe 1 or 2 that we can detect. So what is the actual function of H2AB? So maybe we're not looking for the right phenotypes. And this is where our approach to biology through the lens of genetic conflict was really helpful in this project. Indeed, what we hypothesized was that the rapid evolution of H2AB was the consequence of an ongoing genetic conflict that must be shaping some aspect of placental mammal reproduction. And our new goal was to try to understand what this conflict was. The first conflict we considered was one that deals with competition between sperm cells at the exit of meiosis, and in this case is referred to as meiotic or post-meiotic drive. You can envision how, and there's been demonstration of such conflict occurring in placental mammals. For instance, in our case, H2AB being linked to the X chromosome, we envision X versus Y bearing sperm maybe competing during spermatogenesis or reproduction, and that conflict driving the rapid evolution of H2AB. So this conflict and this competition between alleles in the context of meiotic and post-meiotic drive should affect the completion of meiosis, spermatogenesis overall, and sex ratios, as this is likely the selective force that is then sort of counter selecting for a suppression of drivers that skew sex ratios in this context. So we investigated H2AB impact on spermatogenesis, and we found that there was no alteration in, for example, the establishment of meiotic crossovers during recombination. Daily sperm production was identical between wild-type and knockout males, and sex ratios showed no obvious biases between in crosses involving wild-type males or their knockout siblings across to wild-type females, as shown here. Those ratios were nearly identical. So this suggested to us that H2AB does not affect testis function or sex ratios, and so that made this hypothesis of meiotic or post-meiotic drive of an X versus Y conflict in this context quite unlikely. We consider then another conflict that is known in mammals, for example, which is one that is more a post-zygotic drive, and in this case involve parental effects in the zygotes. In this conflict and in this context, sorry, and this conflict arises where when parents have opposite incentives during reproduction. A classic example of this conflict that influences zygote viability is genomic imprinting in placental mammals and flowering plants. It is essential for development, and indeed the chromatin modifications of germ cells in the context of genomic imprinting is essential for development. And this is really a conflict and an antagonism for resource allocation to the offspring. Those chromatin modifications are going to alter how much resources the zygote is gonna take from its mother during reproduction. And of course, fathers can hijack this process to increase the amount of resource allocated to their pups, while mothers might try to dampen this effect because they want to have multiple litters or maybe mate with other males. So this type of parental conflict should have consequences that are measurable as parental effects coming from the parents. It should affect embryo viability and embryo growth. So in the context of our mutants, we began to investigate those questions. What I'm showing you here is the viability post-implantation by looking at the percentage of resorbed or severely delayed embryos we can measure at about 2/3 of the way in mouse gestation. What we found was that in crosses involving wild-type animals or knockout males or knockout females, there was no difference, a mild increase here with knockout males in the proportion of those inviable embryos. However, the first surprise came when we look at those results in the context of a knockout cross involving both knocked-out parents. In this case, we found that about 1 out of 10 embryos did not pass implantation. So that was quite interesting. From this, we concluded that H2AB loss affects embryonic viability. Another consequence of the loss of H2AB was on the surviving embryos. So not those that died post-implantation, but those that survive implantation. What we found was that H2AB also impairs embryonic growth. Let me show you how. We measure this by measuring fetal weight, so the embryo proper, and placental weight, so extra embryonic tissues in placental mammals. Again, here, I'm showing you this in the context of those crosses involving knockout males or knockout females or both knockout parents. And what we found in this context was that in crosses involving in complete absence of H2AB, if you will, you had about a 30% reduction in both fetal and placental weights in those crosses. And this was true for both male and female embryos, although I'm just showing you here male embryos for clarity. So because this is happening in the context of a knockout cross, this could be a parental effect, this could be a zygotic effect because those embryos are mutant, or maybe a combination of both. So we went about deciphering these contributions using genetics and really using the power of genetics in this case. So to investigate H2AB putative paternal effect, we had to compare embryos that were genetically identical coming from mothers that were genetically identical as well, in this case, knockout moms, and comparing the weights of those embryos only switching around the genotype of the parents, of the fathers, sorry. In this case, wild-type males or knockout males. So this is what we did, and in this case, what we found was that only when the father is mutated for H2AB do you see an effect on embryo, on fetal and placental weights, so over the whole embryo, and this is highlighted here. So this indicated that H2AB is a paternal effect gene, whereby the paternal genotype and not really the zygotic genotype in this example has an influence on embryonic growth, and this is the first reported example of a histone variant with such function in mammals. Maybe more surprisingly, we also found that H2AB was a maternal effect gene. We used a genetic trick, this time keeping paternal genotype constant, comparing embryos of similar genotype as well, but switching around maternal genotypes. And in this case, we had to use heterozygous females because knockout females will only produce knockout crosses in this setup. And so we can't really compare that with wild-type mothers. In this case as well, so here we're measuring fetal weights and placental weights, we also found that when switching maternal genotypes around, so when the mom is mutated for H2AB, do you see an effect on fetal growth, highlighted here, comparing those two crosses. But quite surprisingly, we found that there was no effect on the growth of the placenta in this context, indicating that the maternal effect and the paternal effect of H2AB were somehow quite not fully overlapping. We also ruled out the zygotic effect by comparing the weights of zygotes that were genetically different, so mutants versus wild type, but coming from similar parents, and we found no difference, strongly supporting the idea that the zygote genotype does not influence this phenotype. So I told you that H2AB is a parental effect histone variants, and so our hypothesis was that somehow, because H2AB itself is not inherited to the embryo, somehow paternal H2AB and maternal H2AB leave some kind of an imprint in the gametes that is then influencing embryonic growth. The first question we had to answer was, what was this maternal effect that we saw, considering that H2AB was described mostly in fathers? We found that there was a mild but specific expression of H2AB at the onset of meiosis in females, and this is data taken from this publication here. Meiosis occurs during embryonic development in females, so these are the embryonic stages that we investigated, and we find this sort of induction of H2AB at the onset of meiosis, just like what we see in males. So now really where we're spending most of our time today, and really the big questions that we're investigating in the lab right now is, what is the nature of this imprint? And what is this imprint doing into the embryo following fertilization? So what is the mechanism? Part of that work started in this work I just mentioned, and through collaboration with Derek Jensen's in Steve Hennicoff's lab, right downstairs from us when I was a postdoc. What we decided to investigate was the overall chromatin structure of postmeiotic germ cells, where we know H2AB is still present in the chromatin. For this, we use auto-cut-and-run directed against K4, K27, and K9 trimethylation marks. For those of you who don't know what cut-and-run is, we're going to target PA-MNAs to antibodies that recognize those marks in the context of the nucleus. Because this is an MNase-based assay, this generates fragments of different length. The first type of fragment length that we find in those assays corresponds to nucleosome size fragments, while the most shorter or smaller size fragments correspond to partially unwrapped nucleosomes in the context of our study. What we found was that universally across all of those three marks, we could identify a shift from an accumulation of smaller fragments in our knockouts as compared to the wild type shown here. Knockout is in red and wild type is in blue here. So consistent shift across all three marks, indicating that upon H2AB loss, there's an overall change in chromatin structure that seems to correspond to a partial unwrapping of nucleosomes in this context. Now, it is fine to find differences in histones and nucleosomes, in nucleosome structure in haploid germ cells, but in order for H2AB to have this effect, this inheritance effect, it has to survive the major chromatin remodeling that occurs during spermatogenesis. There's first transition proteins that are interacting with nucleosomes in this context, and then replacement of those structured by protamines almost genome-wide, with the exception of 4 to 10% of nucleosomes. So really, we're focusing a lot of effort today to try to understand how the loss of H2AB is going to affect chromatin in mature sperm, because this is going to give us a hint at what's going on following fertilization. So now I couldn't resist to give you a little bit of an overview of the work in progress. Now that we've established our mouse colony here in France, so that took us a while, that my lab is fully equipped with centrifuge and other amazing tools. This is the work of Joris Mordier and Nine Fress in the lab. We did ATAC-seq in mouse epididymal sperm. And so what we find is that there is a shift in fragment length in those experiments, indicating that there might be structural changes that occur in mature sperm between knockout and wild types, as shown here in this shift that you can see here and over here. What we're doing now is trying to identify specific regions where those changes might be occurring. For example, here I'm showing you our ATAC-seq signal for wild type and knockout animals, wild type and knockout sperm, sorry, to be more precise, over position nucleosomes that were detected in this study, also in mature sperm. And we can identify specific region that might lose or gain signal depending on our genotypes. And so what we're hopeful to identify in the coming months and years is where these changes are occurring and applying more specific techniques such as cut and tag and cut and run on a mature sperm to identify where and how those modifications are occurring. Now, really one other central question that we're addressing to the lab, and we're gearing up now the lab, we have been gearing up for doing those experiments since we started, is really what happens to those imprints, if you will, still to be determined, but what happens post-fertilization and how the loss of H2AB in this context is going to change chromatin modification or gene expression. So stay tuned, because we have all of those tools now with collaborations within the Institute and outside of the Institute. So this is really something we're having a lot of fun doing right now in the lab. So really to come to a conclusion here, I showed you today, so I have this joke about a take home and how you're already home, so that's fine, but I won't do that today. Instead, I will just give you an overview of what this study sort of taught us. I've showed you that we uncovered that H2AB had this parental effect on embryonic growth. And so what I showed you was that these effects were unequal between fathers and mothers. And so we think that H2AB might be involved in mediating part of this parental conflict during placental mammal radiation. And so maybe it is not that big of a surprise that a histone variant such as H2AB and other short H2As arose in the last common ancestor to all placental mammals, where in utero development is widespread and exclusive. Perhaps it is not also that surprising that H2AB is universally encoded on the X chromosome, considering that it can be subject to those opposite or antagonizing selection in male and female, as alleles of H2AB on the X chromosomes are segregating in the population in both sexes. So really, we came full circle. We had an evolutionary analysis that uncovered this unprecedented level of diversification, going all the way to phenotypes that involve reproduction, and sort of having this somewhat satisfying explanation for this evolution is really something quite a thrill for evolutionary biologists and chromatin biologists like me. So now we're spending a lot of time uncovering this. Maybe I have 1 minute left, so I will say that outside of its role in development, we can maybe learn something about H2E function in human. And really, just to put one slide out there, in a paper that came out not too long ago, we showed that H2AB and other short H2As naturally display substitutions that are usually found when H2A is mutated in cancer. And these are known as oncohistory mutations. For example, here, this position, H2AB naturally has a Q here, but this Q is present in a wide array of cancers as a substitution in core H2A. And in this paper, we show that a wide array of cancers do upregulate short H2As, H2AB in particular. And in those tumors that do upregulate H2AB, you have specific signatures of transcription and splicing that are quite interesting to follow up on today. So with this, I think I'm running slightly out of time. So I want to thank, obviously, my postdoctoral lab and thank Harmeet for his amazing mentorship and for the amazing time I had in this group. Janet Young, Anna Wood, Selena Kinley, and Mike Eichbush for the specific contribution to this project. It was amazing to work with those guys. our collaborators at UW and elsewhere, my friends from the Hennicuff Lab, with whom I had a great time. About a year ago, I was still there. Our funding in the US and in France, very important. This is where the money's coming from. This is what allows us to do our job. And I hope I highlighted the contribution of Marine Fraisse and Joris Mordier, specific contributions to those projects today, and as well as I mentioned the students earlier. So I will stop here, and I'll be happy to begin a discussion here and at the coffee chat. So thank you for your attention.
Thank you, Antoine. That was wonderful as always.
Thank you.
I will start with an announcement that was posted in the Q&A as well as in the chat in case you missed it. The EMBO workshop on histone variants for 2021 will be happening in September, and registrations for abstract submission are still open for 3 more days. So the link was posted by Mark Bushbeck in both the Q&A and the chat, so feel free to look in there. And like he says, you can meet other weird histone people. So join us. I've applied too. It's
a great meeting.
Yeah. All right, so we have a couple of raised hands. So I'll start with Pallavi Mustafi.
Hi, Anthony. It was a wonderful talk.
Hello.
My question is very basic, is that this variance of H2AB, do you think in terms of its nucleosome binding affinity or their propensity to find the dimers will be different across male versus female, and will that also like their nucleosome eviction rate, the nucleosomes formed by these variants could also explain the kind of functional difference that they might have?
Yeah, thank you for this question. There is plenty of work on the consequences of incorporating short H2As and H2AB in particular in the nucleosome. Structural work, biochemical work. You have destabilization of nucleosome structures. You have exchange with core H2A that has been studied. So I think that sort of covers some aspects of your question as far as structure, something I didn't mention here. Mm-hmm. Your second point was that, is there a reason for us to believe that there's a difference in the consequence of H2AB incorporation between males and females, if I understand?
Yes.
This is an unknown. We know that in the context of spermatogenesis, you have all this chromatin remodeling. So obviously there's going to be effects, or at least I'm assuming or hypothesizing that there are going to be effects that are going to be specific to males, okay? As far as females, what's going on for the incorporation of H2AB is completely unknown. What sort of replacement might be occurring in females, unfortunately, is completely unknown, but this is obviously something we're looking at now. So there's sort of balanced answer, maybe unsatisfying to you, but so yeah, I think that there can be differences. I cannot tell you right now what the nature of those differences might be.
Yeah. Okay. Thank you.
Thank you for your question. Thank you.
Yeah.
All right, so I am also gonna unmute Stephen Krobit. Sorry if I butchered that.
So looking at the participants.
That's great. Can you hear me?
Yes.
Perfect. Mm-hmm. Wonderful talk. So a couple of quick questions. Have you looked for the binding of your short H2As or Bs on at CTCF sites? Because that's where most of the histones are actually positioned in both mouse and human sperm.
So we haven't looked at CTF sites specifically in the context of our, for example, chromatin profiling in sperm right now, but it's definitely something we will be doing. So thank you for that. We have looked at some enrichment for specific motifs, for example, in the context of the global structural changes we saw in haploid germ cells, so at the exit of meiosis. And we couldn't identify specific motifs in this context, but keep in mind that those changes are widespread. So maybe we don't have the specific resolution to answer that question now.
So in terms of those motifs, you might be interested in a paper by Graham Johnson.
Yes.
In Scientific Reports, which actually goes into some of those binding motifs that are well positioned.
Yes. Okay. Thank you.
The other question I had is in terms of the sharing of transcripts during spermatogenesis by the cytoplasmic bridges. So does that make the X-bearing and the Y-bearing sperm basically equivalent to the positioning of your short histones?
What specific part about the positioning did you ask?
Yeah, so I'm very interested in terms of whether if you had an X-bearing sperm or a Y-bearing sperm,
yes,
whether the position of your variant would be the same.
Yes.
You don't mind.
Okay, so in the context of meiosis, H2AB induction and chromatin association, and this is a great work from David Trematek's laboratory. We found, it was shown, sorry, that H2AB is present during meiosis and after meiosis. And so there is no obvious reason to believe that there would be differences in H2AB incorporation in Y versus X-bearing sperm. Now, for other short H2As that might be expressed later during spermatogenesis, maybe that's a different answer. But so far, we found no differences. So you're right about the cytoplasmic bridges, even for those that are expressed after meiosis. It will not change that.
Great. Thanks so much.
Thank you for your questions.
All right, so we have 2 questions in the Q&A. We'll take those 2 and then move on to coffee chat. The first is from Peter Sarkis, and he says, really interesting talk. Although there is no decrease in the median fertility, it did seem like the H2AB knockout had more crosses that resulted in no offspring. So is this a real effect? And does this indicate failure of fertilization in some of the H2AB knockout animals?
Correct. So thank you for this question. I do have slides that I had prepared for that. What I can tell you is that indeed there is this change here where we have more litters with dead pups, or so like perinatal death. So that's a good observation. This is another place where male versus female H2AB knockouts are going to differ. So that's also very interesting. You have this effect on litter survival in males. You don't have this effect of perinatal mortalities when you use knockout H2AB knockout females. So again, interesting here to consider in the context of the conflict and the antagonism. What we found is that in the context of those assays, matings can occur, yet pregnancies ensue at lower rates. Maybe going with this idea of post-implantation defects that we see, mortality is increased, and this is something you see here. But I think your question was probably more related to that point as well, whereby we can see that, sorry, that's not the right slide. Well, I don't think I have the slide here, but what we investigated in the paper, and I invite you to check it out, is that we don't see a difference in the progression of fertilized eggs going and moving all the way up to the blastocyst stage in vitro fertilization assays. So I think that's to answer directly your question. So yes, so thank you for pointing that out. I think it's another interesting difference.
Yeah, great. All right, last question. How much H2AB is retained in sperm? And is the fraction lower or higher than for other H2As? And this is from Marcus Bushback.
So in fact, H2AB is, as far as we can tell by mass spec, and this is again publicly available mass spectrometry data sets, we cannot detect H2AB in mature sperm. So its effect has to be an effect that it has at some point after meiosis or during meiosis on chromatin structure. And this effect on chromatin structure has to itself sort of propagate onto other chromatin changes in mature sperm. So this is a place where we're currently investigating those changes, as I was showing in the talk. But so far, I can tell you that there's a lack of evidence that H2AB is present in mature sperm. So unfortunately, I cannot tell you the fraction in nucleosome, but that's
and is it question. Right? So you can't compare it to other H2As either, I guess.
So it appears that in mouse, there is evidence that some H2ALs, so these are a different family of short H2As, are retained in mature sperm. Now, as far as the fraction of H2A versus H2AB at the after meiosis, for example, we found that this fraction might be quite abundant. And I want to sort of highlight this by this sort of shift that you see in size here. We think that in the wild type, you have those H2AB containing nucleosomes here, and in the knockout, you have those nucleosomes that cannot contain H2AB in haploid germ cells, and you have this sort of shift here. And so maybe here, the area under the curve here gives you an indication of the sort of amount of histones that might have H2AB. But again, it's a very indirect way of measuring this. And this is just for the pleasure of discussing this question with you guys.
Great, thank you. All right, so that is all for today. If you want to stay for coffee chat, please let us know in the chat. And if not, we will see you in 2 weeks.